So let's get this webinar recorded. Hi, everyone. Thank you for joining. I think we are expecting more than, how many you said, Jesse? 170 registrations. So we're going to be a very popular webinar. My name is Thierry Lacaze. I'm the scientific director of MyCERN. I'm also the nominated principal investigator for the rare pediatric rare disease clinical trial and treatment networks grant just funded by CHR as part of the high-cost drug strategy for rare disease. And that has given me the opportunity to work very closely with Beth and Pranesh over the last almost 1 year now, even before. And this webinar is organized conjointly by Impact and Rare Kid Scans, and we have the pleasure to have two outstanding speakers today to talk about the role of registry in the rare disease space and why it's critical, for the research in patients with rare disease. Maybe first, let me just acknowledge, our lands, and I will, work from a national perspective using the Impact land acknowledgements. So I really encourage everyone here on this call, and we are all Canadian or visitors to Canada, to learn about the indigenous people of the lands on which they live, work, or visit, and we become engaged and allies together. We as Canadian, in the spirit of truth and reconciliations, respect the self-determinations of First Nations, Métis and Inuit, and their right and responsibility in culture, language, and the pursuit of wellness. Now, more from a local perspective, since Beth and Pranesh, speak from Ottawa, and I am actually speak from Ottawa today, I would like also to pay respect to the Algonquin people, who are the traditional guardians of our lands here in Ottawa. I think we acknowledge all their long-standing relationships with this territory, which remains unceded. We pay respect to all Indigenous people in these regions from all nations across Canada who call Ottawa home. And we acknowledge the traditional knowledge keepers, both young and old, and we honor their courageous leaders, past, present, and future. Having said that, I would like to introduce Beth Potter and Pranesh Chakraborty. Pranesh is the current new chief and chair of the departments of pediatrics at CHIO, and he has been at CHIO for now more than 20 years, working as a clinician for patients with inherited metabolic disorder. In 2006, he led the establishment of a newborn screening Ontario at CHIO, and he was the medical and laboratory director until he became the chief chair of pediatrics last year. He has done tremendous work in regards to advocacy at the provincial level, but also at the national level. He's actually member of the implementation working group of the strategy for rare disease. And Pranesh will be joined by Beth Potter. Beth is professor of epidemiology and public health at the University of Ottawa. She is a research chair in health service for children with rare disease. And we all know Beth as the lead and the principal investigator of Inform Rare. Inform Rare was founded by CHR a couple of years ago, maybe 3 years ago, and has done a very impressive work in helping the establishments of guidelines for registry-based clinical trials using 3 conditions, and I'm sure she will, mention and go into detail on Inform Rare, which is really an outstanding example of a very successful network in the rare disease space. Inform Rare is recognized as an ACT network, which is also an example of the leadership of BED, because it's a privilege to be part of ACT. And BED is now co-leading the sub-platform on registry on rare kit scans. So they will both present today on a clinical trial in rare disease, the role of patient registry. And I think you are first to start, Beth, correct? So I give you the floor. Thank you.
Great. Actually turning things over to Pranish to start. Whoops.
Yeah, thanks. Thanks, Yaki, and thanks, Beth. If we can go to the next slide. Yeah, Beth and I are going to go back and forth a couple of times, through the presentation. And, Thierry, thanks for the introduction and actually pointing out how long Beth and I have known each other. We both came to Ottawa right around the same time, met on the ground floor. And I think I was smiling during the introduction because a lot of the work that we're going to describe here has really started right after we met. Trying to understand how we can improve care, improve knowledge, and improve outcomes for patients with rare disease and specifically inherited metabolic disease, both as a specific disease area, but also as an example and an exemplar for how to approach some of these questions in rare disease. So, this is maybe 20 plus years in the making to where we are now, probably another 20 years of good work to do and beyond that. So our objectives are, as noted here, describe challenges in generating high quality evidence about treatment for rare disease, summarize some of the strategies proposed to address these challenges and emphasizing the potential value of patient registries specifically, describe features of registry-based randomized clinical trials and discuss scientific and practical challenges associated with such trials, and then summarize some current guidance and emerging developments in designing and improving rare disease registries to support clinical trials. And also notice in the participants, we have a lot of members of our team here, including members who lead particular aspects of this work. We have an acknowledgement slide at the end that lists a lot of these people, but this is a big team. It's a well functioning team. Everyone's bringing very important, specific expertise, including on things like methodology, things like patient engagement and patient research, et cetera. So we're just presenting on behalf of this group, and happy to see many of them here. So next slide, please. Sorry, we're still having a lot of birch pollen in Ottawa, and I'm very allergic to birch pollen, so my antihistamines are helping, but I will every so often have to pause. So rare diseases, definitions of rare disease vary, and one of the hardest things is to once you get to the nub of an issue is to actually be on the same page as what you mean. The concept of rare disease is intuitive. But it implies something about prevalence, but there's no real fully agreed upon standard definition. Often it's defined as a prevalence of less than 1 in 2,000. But in aggregate, and I think Durhane Long Reader from Ford always does a good job of emphasizing this, in aggregate, people with rare diseases make up a significant part of the population and the population of people with an ongoing or chronic condition that needs treatment. The majority of these are single gene disorders. Their children are often most impacted, but it is not solely an issue of childhood health and pediatric care, but they are most impacted. There's often high morbidity, and many rare diseases are progressive, and that has impacts for patients, caregivers, and the healthcare system as a whole. Some, I might say many rare diseases are associated with the risk of mortality or reduced life expectancy. That contributes to their prevalence and impact in child health, and it's important to remember. New disease modifying therapies that can alter natural history, improve outcome, and are emerging. There have been some very established therapies, but the pace of new therapy development and coming to market has really has really accelerated. And, one thing to note is that once you're treating a disease, especially a disease that had been perhaps in early infantile or early childhood fatal, you can see emerging parts of the disease in the treated population. And or to give many examples from the inherited metabolic disease world, it's a question that comes up in spinal muscular atrophy and Pompe disease, another, muscular disease that's an inherited metabolic disease. Now that there are very effective treatments, we're seeing treatment alter natural history as well. Okay, next slide. Okay, this is just a visual representation of what I already noted. One is that there's an accumulating number of new diseases that have been recognized, understood, in the process of being understood. And so there's a recognition of new diseases, and Canada has been in the forefront of a lot of the work, that Kim Boycott has really led in terms of understanding how we discover and delineate new diseases using genomic technologies. But at the same time, we're seeing that a very similar growth in new therapeutics. This is a graph based on orphan drugs, but I think it's also important to remember that treatments go beyond drugs, and there's many additional modalities of treatment that are also growing in terms of availability and impact. Okay, next slide, please. Okay, so all of this does lead to policy issues, crystallizing around this term of drugs for rare disease, and sometimes talked about or wondered about, is it, drugs are very important, but those other therapeutics are also very important, and all that pathway and system of care involved in for people with rare diseases, all circle around together. But it's kind of crystallized around this concept of drugs for rare diseases. So some of the challenges include rigorously evaluating the effects of drugs for rare disease. Many are expensive and have high per patient costs, and there are important impacts for patients and healthcare systems around both these aspects of drugs for rare disease, but also the pathway of care, the system of care, recognition of patients, avoiding diagnostic odyssey, population-based screening, et cetera. So this impacts many areas of health policy decision. So, the good news that, we are all very excited about in the rare disease community, was Canada's National Strategy for Drugs for Rare Diseases, announced in March of last year, and investing $1.5 billion over 3 years to promote a few things. National consistency and coverage for drugs for rare disease, including supporting improvements in screening and diagnostics. So it was really good to see some of that recognition of the whole system of care. Support patient outcomes and system sustainability with a national governance structure. Improve collection and use of real-world evidence, including from registries, to support clinical and policy decisions. And Beth had bolded that here, and I think that was an important bolding to have. And then advancing research to support improved diagnostic tools, population tracking, and rare disease clinical trials. NSTA noted he's a nominated PI for Rare Kids Can, which is an important part of this work. Okay, next slide. Okay, this is a slide that I thought I would just share and trying to show, how some of those parts of the system of care, interrelate. I drew everything here with straight lines, and some of these lines are straight. I already mentioned the term diagnostic odyssey, so we know some of the lines are squiggly or iterative or circular, unfortunately, sometimes. But they're just here again showing how you can have population-based screening as a way to have timely recognition of a patient early in their natural history of the condition so that they can access treatment in a timely way. In diagnosis, you have a symptomatic patient who needs somehow to get to clinical assessment. That's often where the squiggly line happens to get to that first clinical assessment. Then you can have a second squiggly line getting to a subspecialist assessment. Then you can have another squiggly line getting to a diagnostic testing pathway. but all with the goal of trying to get treatment diagnosis early enough in the natural history in the course of disease to be able to change the outcome, and obviously also to avoid that diagnostic odyssey and the impacts that directly has on people who experience that. And then, increasingly we have this, stream of treatment and ongoing care where, you can have treatments of multiple modalities, drugs, transplantation medicine, surgeries, nutraceuticals, and, medical foods, stem cell therapies. And treatment often doesn't occur in isolation and always has to be considered in the system of care for providing those treatment and ongoing care as well. And then there has to be a cycle of monitoring, using various outcomes, including, very importantly, patient-reported and patient-prioritized outcomes, laboratory assessments, adjustments to the treatment plan, and then an ongoing cycle of assessment and improvement of the treatment plan for the person affected. So with that, I think I hand off to Beth next, in terms of talking about some of the evidence challenges and how we might, approach that.
Great. Thanks, Pranish. And so those of you who are researchers in the field of rare diseases will be well familiar with these evidence challenges. And so just present them fairly briefly. And for each, I have one or a few quotations. And the quotations are drawn from the work, the PhD dissertation work of Kylie Tingley, a student that who Pranish and I worked with a few years ago, who interviewed patients and caregivers, policy advisors, and healthcare providers about evidence around therapies for rare disease. So the first challenge here is that for many rare diseases, we have sparse evidence about their natural history, combined with a large amount of heterogeneity across patients who have the same rare condition. And this is important, of course, because we really do need to understand natural history in order to know whether interventions are going to appreciably alter natural history. I won't read these quotations, but just give people a little bit of time to kind of look at them as I'm talking. So the second challenge inherent to rare diseases and their definition is that it's difficult to generate strong evidence of treatment efficacy given small patient populations. And I think this is widely recognized by patients and caregivers, policymakers, and physicians. And we'll talk very briefly about sort of some of the strategies around that. The third challenge is that evidence of treatment efficacy, when it does exist, may not translate to real world patients in routine care settings and over longer terms than relative to the sort of selected subpopulations and shorter durations of typical efficacy trials. And this challenge is really, it's not specific to rare diseases. I mean, we see this with across healthcare, but I think arguably it's compounded in rare disease by the earlier challenges. So the challenge with understanding natural history and also some of the limited efficacy evidence. And then treatments are often not evaluated using meaningful, harmonized, and well-measured outcomes. Again, not a challenge that's specific to rare diseases, but because we have such limited research resources, this is part of trying to make best use of those resources, which we'll talk about quite a bit over the seminar. And here you can see a quotation from a patient or caregiver talking about measuring really what's important and a healthcare provider as well. So some of the strategies that have been proposed to address these evidence challenges, I think, fall within some sort of broad categories. One is bringing people together. So multi-center networks that can bring small populations with rare disease kind of together, working together in the same study to sort of increase sample size, but also the researchers, the providers, the policymakers, and patients and caregivers. So kind of everyone working together. And I think that the Rare Kids Can Network and the funding announcement around that is a great example of how we'll be able to move the field forward as a group. use appropriate study designs. And so, of course, we need to maintain high evidence standards and lots of work around developing clinical trial designs that are more suitable when we have small sample sizes. And I would add to that that we also need to still be thinking about pragmatic designs and routine settings and some of that effectiveness evidence as well. And then investing in high quality infrastructure. And so we're going to be talking mostly during the webinar about, in general, and then with reference to some of our own work, about patient registries as a source of longitudinal data that can help to support evidence generation for rare disease therapies and the role of core outcome sets. So a patient registry is an organized system that uses observational study methods to collect uniform data to evaluate specific outcomes for a population that's defined by a disease, condition, or exposure, and serves one or more stated purposes. This definition is widely used and I think is important to think about as we go through sort of thinking about all of the guidelines and methods around the use of patient registries, particularly concepts around uniformity of data collection, specified outcomes, and considering purpose. A disease registry is a type of patient registry whose members are defined by a particular disease. And so this is really to distinguish a disease registry from a product registry, which would be focused around building evidence for a specific medicinal product, one drug, for example, whereas a disease registry has a broader focus. So patient registries are a source of real-world data, and this is really what I think has attracted the health policy community in particular to thinking about patient registries as a strategy around some of the evidence challenges that we talked about. So just a couple of definitions here. Real-world data is defined differently in different sources, but typically refers to individual-level data about patient health status or healthcare delivery. And the defining feature really is that it's collected outside of randomized controlled trials, at least explanatory trials. And real-world data is often generated during routine clinical care. It can be generated from various sources, including disease registries. Real-world evidence then is really evidence that makes use of real-world data to understand the use, benefits, or risks associated with a medical product. And so here, this is another kind of important distinction, I think, between real-world data, for example, the patient registry, and then real-world evidence, which is whether that registry, those registry data are suitable for the purpose for which they're being used. So in terms of thinking about registries versus other sources of real-world data, registries may include data elements that are less likely to be available in some other sources of routinely collected data. For example, data generated healthcare administrative data or claims data, which may be less likely than registries to include patient-reported outcomes and more in-depth data about indicators of disease severity. On the other hand, there might be more concern about registries as a source of real-world data around their representation of the target population. So, for example, registries are typically rare disease registries. Most of them are consent-based registries, and so we have to think about recruitment and retention and whether they reflect the underlying population. So again, just needing to really think about the purpose, both of the registry and how it will be used. So if we think about how patient registries can contribute to clinical trials for rare disease therapies, I think we could consider sort of three broad categories of purpose here. The first is to characterize natural history to inform clinical trial design. So registries generating natural history data to describe the distribution of outcomes among rare disease patients, understanding the progression of rare disease over time with age, for example, and differences among subgroups, evaluating and selecting potential endpoints and validating endpoints for clinical trial design, determining eligibility criteria, and informing sample size calculations. A second purpose is that registries can be used to provide a platform to directly facilitate clinical trial conduct. So at the simplest level, but critically important, is using registries for recruiting participants for clinical trials. So being able to actually understand how many individuals are may be available to participate in trials and being able to reach out to them. Registries can also be directly, or clinical trials, sorry, can also be directly embedded in registries. And so registry-based randomized controlled trials, which I know is of interest to some folks on the webinar, we'll talk about in a little bit more detail. And then registries can also be used to provide long-term follow-up data for clinical trial participants, even if the trial hasn't been embedded in the registry, but the trial data could be linked afterward. And then finally, registries can be used to augment data from clinical trials in order to inform policy recommendations and decisions. So thinking about health technology assessment and also regulatory processes. There's a lot of interest in the rare disease community around the potential use of real-world data from registries as a source of external control data for single-arm clinical trials. We're seeing a lot of single-arm clinical trials, particularly with new disease-modifying therapies like gene therapies. And I think methods around use of registry data to provide a source of external controls are going to be really important, and I'm sure will be taken forward with Rare Kids Can. And then registries can also be used to generate real-world evidence about safety and effectiveness in more routine patients and settings and in larger patient populations and over longer follow-up time periods than is possible in clinical trials. And this in particular, I think, is of great interest to the health technology assessment and reimbursement policy decision-making realm. So a little bit about registry-based randomized controlled trials, which is something that Inform Rare has really been interested in over the past few years. Interest in this, in RRCTs, I think really took off after the publication of this particular commentary, which was a commentary on the TASTE trial, a cardiovascular trial that was embedded within a Swedish heart disease registry, the SwedeHeart Registry, which is a clever name, I think. And the SwedeHeart Registry, I think, has been used since for other registry-based randomized trials. But this commentary was really reflecting on how the trialists were able to rapidly recruit thousands of patients and conduct their trial at a relatively low cost, compared to a similar trial that would be done outside of the context of a registry. And so commented that this represents a disruptive technology that transforms existing standards, procedures, and cost structures. And commenting really on how this particular example seemed to have the best of both worlds. So the benefit of the randomization to reduce bias and maximize internal validity, but also the benefit of external validity and efficiency from the use of the registry, the real world data. However, despite arguments for the suitability of registry-based randomized trials for rare diseases, a scoping review that Inform Rare conducted, which was led by Martin Afringa and Ami Baba and colleagues, found that RRCTs are rarely used in rare disease settings. And from that scoping review, RRCTs are typically pragmatic rather than explanatory trials. So that may be part of the explanation. They're also used broadly, most commonly in cardiovascular and oncology trials, which makes sense because of existing registries and population-based registries and sources of data. And they're far more common in Scandinavia than elsewhere, probably because of the availability of population-based registered data. So the TASTE trial, for example, their primary outcome was mortality, but all of their secondary outcomes, some of which were sort of clinically complex, were also collected based solely on registry data. So briefly, a registry-based randomized trial, this is sort of an overview of the approach. So you could imagine a patient registry where you have ongoing collection of data on outcomes, treatments, and other characteristics. You could evaluate who's eligible for a clinical trial. Those who consent to trial can be randomized. There's no particular trial design that's specific to the registry-based randomized trial. It's really just the concept of a trial embedded in a registry. So there are even cluster-based, cluster randomized trials that are embedded in registries, for example. You would have potential registry participants who decline to participate in the trial, but may consent to remain in the registry, and you may have ongoing collection of data about treatments they receive and their outcomes. Then you would have the trial participants who continue. Registry-based randomized trials can incorporate trial-specific data collection. I think in our scoping review, over 2/3 of registry-based trials collected the primary outcome based on registry data, but many trials incorporated other data elements that were specific, that were trial specific. And then you would have some registry data on people who withdraw from the trial but consent to remain in the registry, and ongoing collection of registry data. So you can see here some of the advantages of a registry-based trial in terms of having access to data before and after a clinical trial, and also from registry participants who are not trial participants or who don't complete the trial. So potential advantages of registry-based trials that are in general and specific to rare diseases really revolve around, I think, these two concepts of efficiency and external validity or representativeness. So efficiency in terms of patient recruitment and data collection from existing cohorts and using existing data, having access to additional information pre and post-trial, and for people who don't participate or complete the trial. And then depending on the registry, the potential for high external validity. The limitations that have been described are mostly around, I think, outcome assessment. So depending on what the outcome, the trial outcome is, the timing of outcome collection may be challenging if it's dependent on the frequency with which outcomes are collected within a registry. And importantly, I think outcome data quality in a registry may not meet trial standards, and yet evaluating this is often poorly reported in RRCTs. And then finally, particularly for a new therapy, important data elements may not align with registry data collection, and you lose some of the efficiency advantages of registry-based trials if you're collecting a lot of information from outside the trial. And so really, I think what we see is that both realizing the advantages and avoiding the limitations of RRCTs are dependent on the nature and the quality of the registry. There are reporting standards for registry-based randomized trials. So they're part of the extension to consort routine that covers trials that are embedded in sources of routinely collected data, including registries, cohort studies, and other like electronic health records, claims data, and so on. And there's an extension to SPIRIT for registry-based randomized trial protocols that's currently under development. Also, as part of some of the work with Inform Rare, Martin Afringa, Caroline Krause, and Joanne Tay and their team led work to try to distill from both commentaries and the trials that were part of their scoping review some of the key recommendations that have that we might be able to make in order to take advantage of registry-based randomized trials while avoiding limitations. And they had 10 recommendations, and in their original paper they're described in a lot more detail. So I've kind of rolled some of them up and summarized them here, but there's recommendations for registry designers and administrators to create trial-ready registries, and then also for trialists. So for registry designers, the recommendations include using core outcome sets, which I'll talk about in a moment, to inform outcome selection and measurement. Identify critical risk adjustment variables that are typically associated with outcomes, and that could be incorporated in the registry. Ensure that there's an updated capacity mechanism to identify potentially eligible registry participants and contact them. And then some recommendations around adhering to guidance in general for developing high quality disease registries, including recommendations around governance. For trialists, this is really about, first of all, evaluating whether a registry is suitable for an RRCT. So choosing registries that have a broad coverage of the population, that have longer term follow-up. So thinking about external validity and representativeness, exploring opportunities for further data linkage where possible, implementing quality assurance checks to try to alleviate some of the concerns around outcome data collection within registries and whether it meets the standards for a trial. And then using the registry data to enhance the trial at the design stage. So in terms of things like eligibility criteria and sample size estimation. And then a recommendation for both registry designers and trialists to be thinking about consent and research ethics. So these recommendations are part of, I would say, an almost overwhelming number of sets of recommendations and guidance documents around patient registries, and including rare disease patient registries. There are several sets of recommendations that have been published for improving registries and or evaluating registry quality generally. And so I've just put a link there to the AHRQ guidance from 2020, which is particularly comprehensive and helpful. And then specifically in relation to the suitability, so evaluating whether real-world data from registries is suitable for a real-world evidence need. And so there are there's guidance for evaluating whether registries are appropriate for regulatory decision-making processes, health technology assessment, and specifically for registry-based randomized controlled trials. As some people may be aware, standards specific to rare disease registries are currently being developed by Canada's Drug Agency as of May 1st, so formerly CADIT, in support of Canada's rare disease strategy, and I think will be extremely helpful and important to the Canadian rare disease community in terms of thinking about specifically what registries need to do in order to be able to support real-world evidence needs in Canada. A recent scoping review was published to inform those standards, and I think particularly because it's so recent, is an important resource that synthesizes a lot of guidance that's out there to improve rare disease registry quality. And they've got summarized this guidance kind of within 3 main domains and then a number of subdomains. So the overarching domains are governance, data, and then information technology. And so I'm not going to go through that in detail, but did want to talk a little bit about the importance of common data elements and in particular outcomes. So this slide was adapted from a presentation given by Martin Afringa back in December of 2022 at a consensus workshop that we held as part of developing a core outcome set for pediatric mucopolysaccharidosis. And I think really effectively makes the point about the problem with lack of harmonization in outcome selection and measurement, that when each team measures different outcomes or in different ways, evidence can't be combined to inform changes to practice and policy, which is a failure to make best use of limited research resources, which we really can't afford. We can't afford not to make the best use of limited research resources in rare disease. And so this is not a problem that's specific to rare diseases. And in recognition of the need for harmonized outcomes, there's a worldwide, well, there are several initiatives, but there's a worldwide initiative led from the UK called the COMET Initiative, Core Outcome Measures in Effectiveness Trials, around developing core outcome sets across a whole range of areas of health, where a COS is an agreed minimum set of outcomes, typically 6 to 9 outcomes. I don't know if Martin is here or not, but if he was, I think he would be saying closer to 6 because there's concerns about feasibility. And we really want, the purpose of this is really to hone in on what are the most important outcomes as agreed by multiple interested communities and really promote uptake of measurement of those outcomes. So a minimum set of outcomes that are recommended for inclusion in all studies for a specific disease or area of health, for clinical trials, but also for observational studies, including registries, and for use in routine care. And so, as I mentioned, the premise is that these outcomes really need to be agreed upon by multiple interested communities whose perspectives needs to be considered and incorporated. The methods for core outcome set development typically include literature reviews and consensus methods, for example, Delphi surveys and consensus workshops. Patient engagement has been increasingly recognized as critical to ensuring that core outcome set development includes patient and caregiver perspectives effectively. So how important they consider particular outcomes to be. And I think importantly has been identified as an area that's in need of improvement from a review of pediatric core outcome sets led by Nancy Butcher and Martin Afringa and their team. There are guidelines and standards for both COS development and reporting. And I wanted to just finish talking about core outcome sets here by indicating that I think we're extremely lucky in the pediatric rare disease community here in Canada that leaders in this field for core outcome sets include Nancy Butcher, Martin Afringa, and in terms of patient partnership and engagement for core outcome sets, Maureen Smith, who I'm sure many of you know as part of Impact, Rare Kids Can, and so on. And so I'll turn things back over to Pranish in a moment, but just wanted to first conclude this section of our webinar by summarizing that really, I think in response to unmet needs for evidence to support policy, there's been a recent focus on the potential value of patient registries. As with other sources of real-world data, patient registries vary in their purpose, their quality, and their suitability for answering specific research questions. And I think this has prompted the development of guidelines and standards, which are extremely helpful. But I think there'll continue to be an ongoing need for a case-by-case assessment of whether registry data are suitable or fit for purpose for particular real-world evidence needs. Currently, many patient registries require, including the ones that we've developed, as you'll hear about in a moment, require active recruitment, active data collection across sites and among participants, and are resource intensive. Ideally, we will be able to rely on population-based, routinely collected data while still retaining the details, including patient-reported data that we need to answer important questions. There's been an increased interest in methods associated with using routinely collected data linkage across jurisdictions, and so on, across all areas of health research. And with artificial intelligence, electronic health records increasingly incorporating some patient-reported outcomes, learning health systems, and so on, I think this may all change in the future. So I think with that, I will turn things back over to you, Pranish.
Great, thanks, Beth. So for the next few slides, we're going to talk about Inform Rare and the Canadian Metabolic Patient Registries. Before going to the next slide, I think what Beth just went over really is the theoretical, scientific, and underpinning of our approach to how we have evolved into this current state of our work. So hopefully you'll see that reflected in how we're approaching, how we have approached, and how we are approaching the next steps in our work. So with that, maybe the next slide. So again, for us, the context is inherited metabolic diseases, and that is my clinical area of expertise. For about 1,000 individual inherited metabolic diseases that are rare single gene diseases. They typically involve defects in one or more biochemical pathway. Again, typically they are diagnosed early in life, and I did see that Martin Afringa is in our audience today, but we may have a couple of our adult metabolic disease colleagues in the audience today as well. I mean, I think also increasingly we have people who are surviving to adulthood and with inherited metabolic diseases, but also some diseases that are presenting in adulthood in the same category. But again, typically diagnosed early in life. Clinical manifestations vary from acute illness to chronic multi-system diseases with multi-system sequelae. This area of disease affecting people exemplify challenges of delivering high quality care for children with rare diseases generally. The healthcare needs are disproportionately high. Patient and caregiver impacts are often substantial. Treatment costs to families and to the healthcare systems are typically high. And evidence regarding treatment effectiveness is sparse, but also difficult to produce. Oh, and yeah, one slide. So our first kick at the can at this, perhaps after, you know, 6 years of discussion and planning, was the establishment of a Canadian Inherited Metabolic Diseases Research Network. That was a placeholder name with a placeholder acronym that never went away, so we called it CIMDERN. And it was a multidisciplinary research network focused on rare pediatric IMDs. And it was a collaboration across 13 Canadian metabolic centers. It was established in 2012, and at that time, CIHR had run a competition for emerging teams in rare disease. And I remember well, Beth and I with John Cronick, who was just finishing his stint as chair of pediatrics at Dalhousie, you know, had many conversations about how we could move the needle for patients with inherited metabolic diseases. And you know, while a lot of the concepts that Beth presented have evolved substantially in that time, you know, the problems were, I think, even then very well understood. And you know, there was a recognition of a role of registries and of patient data and of real world data to produce real world evidence that was already circulating at that time. Maybe not with the same words, but with you know, the same problem statement. So this was a major project, and it was a major part of that project was developing a consent-based cohort of 798, so almost 800 children with IMDs who were born between 2006 and 2015. And a retrospective chart abstraction of clinical data was performed, as well as surveys to collect caregiver reported data related to this group of 800 patients. This advanced our understanding of variation in treatment, clinical heterogeneity in disease severity and outcomes, and gaps in care from a caregiver perspective. But this very large and rich accumulated set of data was not well suited to support evaluation of treatment and ongoing care along the lines of what I've presented in the previous slides. And if I go back to that diagram I presented of screening, diagnosis, and treatment, you know, ideally you're minimizing time for recognition of a person with a condition that requires treatment and maximizing their time with good health outcomes in that area of ongoing care. And that's where we also needed to figure out how to best collect longitudinal information organized in a way that spoke to you know, a common and mutually agreed upon set of core outcomes, et cetera. So I'm not going to reiterate all of that slides, but how are we going to spend time in that box in, where a person is hopefully going to spend most of their life. And maybe we'll have one and done treatments, but they will still be interacting with the healthcare system around safety and efficacy of those treatments. So our goal with Inform Rare was to build on the strengths of CIMDERN and seek to address key limitations, especially around data quality and consistency, sustainability, and need to facilitate intervention settings. So this was a partnership of CIMDERN and the Canadian Neuromuscular Diseases Registry, with the goal of having rare genetic diseases for which treatments are available and for which there was a need for longitudinal data to understand natural history and evaluate care. So This was established in 2020 and founded following a receipt where Beth was our nominated PI. Martin and Nancy were part of this. I think I saw Maureen Smith on the call as well as, another co-PI by the CIHR Innovative Clinical Trials multi-year grant. Co-design is a major part of the approach with families and patients, healthcare providers, policymakers, methodologists, and research ethicists being highlighted. The goal is to generate evidence to support decision-making, to improve care and quality of life for children with rare inherited metabolic diseases and neuromuscular diseases, starting with 3 conditions. Phenylketonuria, PKU, the disease where really a transformative treatment was identified as a nutraceutical approach in the 1950s, launched population-based newborn screening as a program in the 1960s, and where we continue to have a need to improve and care both for safety and access to treatment, all the way to improving outcomes, which still has room for improvement. Mucopolysaccharide storage diseases, which are a set of lysosomal storage diseases that really have multi-system involvement, and then spinal muscular atrophy. So, in many places, the newest addition to newborn screening programs, where transformative therapies have really come onto the scene in a big way, and transformative therapies that really need implementation very early in the natural history of disease. So these are the 3 exemplar diseases for the Informed Rare Network. So through prospective patient registries focused on the collection of core outcomes to support observational studies and registry-based randomized trials. So that forms the focus of the goal using these 3 exemplar diseases. Okay, so this is, I think, something that can't be overemphasized. I'm very happy to see Maureen here on the call, and it's really under her leadership as a co-PI patient researcher, patient partner, that patient engagement has been at the core of all of this work. And sometimes I pause on the word engagement. I think patient embedding, patient involvement in every aspect of the research, I think, has been a core part of the work. So again, under Maureen's leadership, it cross-cuts all of Informed Rare's research to ensure a focus on high-priority, meaningful research questions and outcomes, and patient-centered design of our studies. So the metabolic patient registries were developed with co-leadership also with patient organizations. So PAN-PKU Plus as the Canadian PKU patient organization, and the Canadian MPS Society as the Canadian organization for patients with mucopolysaccharide storage diseases. Additionally, patient partners and advisors have contributed to the development of websites, including infographics and videos, consent and recruitment documents, core outcome sets underlying the registries, and selection of questionnaires to measure patient and caregiver reported outcomes. So the registries, again, have been developed as a partnership, and as a partnership between the Canadian MPS Society and CAN-PKU Plus, and I'll have a slide on governance. There were no existing, from the perspective of the patient organizations, there were no existing public patient registries for MPS and PKU, and a long recognized value of comprehensive patient registries independent of specific drug development. It's an important concept as well to provide accurate data on patient populations. And from the Informed Rare perspective, the need for high-quality patient registries to support observational studies and RRCTs, as Beth has discussed, for PKU and MPS. Okay, this is a depiction of our underlying values and registry objectives: sustainability, accessibility, scalability, public benefit to patients and families, and building research capacities, with the following specific objectives that you can see on our website: but documenting the natural history of PKU and MPS in Canada, understand the experience and priorities of patient Canadian patients and caregivers over time, facilitate the design and conduct of randomized trials, provide post-trial follow-up data on longitudinal patient outcomes, and provide a platform to invite participants with consent to other research projects. And finally, also to facilitate sharing of de-identified data with external researchers, again, with participant consent. So I think this is my last slide, and then Beth has a couple others. Thierry gave me a heads up that we have to finish up soon, so I accelerated my speaking pace. So this is a brief depiction of the governance structure to allow the type of work that we've talked about to occur. There is a registry steering committee, and the footnote here, number 1, includes the roles and the representatives within there. So patient organization representative, a CHIO-RI representative, and CHIO Research Institute is the coordinating center and data steward. Hence the CHIO-RI representative, additional patient and caregiver members, additional healthcare providers, methodologists, and ethicists. And again, it's a partnership between the Academic Hospital Research Institute and who, in the future, should there be a PI from another organization, that this role could be transferred to another academic hospital research institute if needed, and the patient organizations. There are funding partners who often are funding directly to the research institute, but that funding could also go to a clinical site or to the patient organization directly and help support it. Our registry platform developer and host is the NORD-IM Rare, so the National Organization of Rare Diseases platform. And data is stored on Canadian servers for Canadian registries such as ours. And participants and clinical sites with the various types of agreements that you see here depicted required between all of these players to create this ecosystem to allow the creation of the patient registry. And I can't overemphasize the need for very robust governance and a lot of care going into this. But once you get here, this is a scalable governance structure that can be leveraged for further work. So I think with that, Beth has a couple more slides, and then.
Thank you, Pranesh. Thank you, Beth. I invite everyone having questions for our 2 speakers to send them to Chelsea, and Chelsea will make sure that those questions are addressed. There are a couple of questions in the chat box, and one is important, I think, from the quality of the registry. So the issue of representativeness, and how is there any guidelines or guidance that allow that the people that actually register or are on the registry represents the population. So the issue of access, equity, and diversity. And can you answer that, Beth?
Yeah, I mean, I think that's a huge challenge, and certainly like incorporated in guidelines for evaluating suitability and fit for purpose of registries. There's information about evaluating representativeness. At the same time, I think what's really needed is to do a better job in all health research, particularly where we're enrolling people from clinical sites, which is often what we need to do in rare diseases. To actually find strategies that better address equity, diversity, and inclusion. Even, for example, with our registries, like a concern that we have is about the web-based data collection, and the registries are available. We didn't get to kind of present. We had a few more slides there on our registries, but the time went by quickly. But there's lots of information on our website about the registries and some of the specifics. And people are welcome to follow up with us if you're interested. But I think there's a huge need in the field of rare diseases. And I think that is something that, to my understanding, that Rare Kids Can is also taking forward, is to look at what resources are available in clinics where people are being recruited into projects like this, in addition to kind of outreach. But we really need to better understand at the enrollment stage and retention stage how to make sure that our research is more inclusive.
Thank you, Beth. I think that we have to stop here, but I am convinced that there will be a opportunity for you trainees of impact and mentors to continue having those conversations either in with Rare Kit Scans or Inform Rare or other opportunities of webinar coming soon. Chelsea, do you have a final words, or Nancy or Lauren for the audience before we close?
Just wanted to say thank you everyone for coming today, and thank you so much Beth and Pranash for this really rich and insightful talk about patient registries. So everybody who's an Impact Fellow, please stay on the call, please stay on the Zoom link for your closed Q&A discussion period with Beth and Pranash. And thank you so much for everybody else who attended today. I think there is some great questions. So maybe Chelsea, we can also put Beth and Pranash's email in the chat if that's okay, if there's any questions for a follow-up, and otherwise we'll wrap up here. This will be hosted online, so you can go back to the recording. We'll send out some information about that later. It'll be on the RISE Learning Management System. Thanks, everybody.